Nucleic acid extraction methods. Nucleic acids are isolated from a variety of specimens: blood, buffy coat, bone marrow, solid tissue, lavage fluids, bacteria, viruses, fungi, organelles, mitochondria. More specimens adequate for amplification by polymerase chain reaction: dried blood, plasma, saliva, urine, bone, teeth, amniotic fluid greater than 8 weeks, hair follicles, hair shafts, buccal cells. Cerebrospinal fluid, formalin-fixed tissue, feces, and soil. DNA extraction yields, so you can see about how much DNA we can get out of each type of sample. And the three main methods of DNA extraction. There's the organic method, which uses organic chemicals, phenol, and chloroform. The inorganic method, which uses inorganic chemicals, detergents, EDTA, acetic acid, and salt, so we salt out the DNA, and spooling. And then the solid phase. DNA is immobilized on a solid support bead or column. The organic protocol is extremely dangerous because of the chloroform, phenol. Those chemicals are really toxic, so we typically don't perform that one unless that's the only method that's available. The inorganic one is good, but it doesn't have as high yield as the solid phase. The solid phase is kind of your go-to method if you're working in more of a clinical setting. DNA being immobilized on a solid support, these come in kits, and a lot of our automated systems use this type of DNA extraction method, but basically if it comes as a kit, you just go through the kit protocol, and in the end, you have really purified, great isolated DNA. So in order from best to worst, I would say organic is the worst, typically only performed if that's like the last option, and uses those toxic chemicals. Inorganic is good. It's your kind of go-to if you're going to do some research or work in an academic lab. You would most likely do the inorganic part of the DNA extraction. And then solid phase is kind of the best. So that's the go-to process that we would use in a clinical setting or in some of our better research labs that have the funds to be able to purchase these specialized kits. All of our automated methods are going to be using the solid phase for the most part. So specimen preparation: bone marrow or peripheral blood, density gradient centrifugation, differential osmolysis, and streptokinase. So we basically have to break apart the cells that are inside of the bone marrow without, and there's different methods listed here for doing that without disrupting the DNA. Tissue: freeze, crush, mince, and enzymatic digestion. So since tissues are multicellular, in order to get to all of the cells that may be embedded like deep in the layers of the tissue, you need to use much more harsh methods to actually extract the DNA. For plants and fungi and plants both have a pretty rigid cell wall, so sometimes they can be tough to crack, but you want to homogenize or vortex with glass beads to get to the DNA. So kind of going through the organic procedure. So here you can see that there's cells in suspension. You would add some type of lysis buffer, which is going to lyse open the cells and expose all of the internal cellular components. Now we don't want anything but DNA left at the end of this procedure. So we have acidification with acetic acid and salt, and that will have all the cellular debris kind of fall out of solution, and those can be, once you put this in a centrifuge, they're all going to kind of go to the bottom, and then your DNA is suspended in aqueous solution. Then you would do an extraction with phenol or chloroform, and then that would separate the organic phase from the DNA that's left in the organic or aqueous solution. Then you would precipitate that with ethanol, and you would pour off the ethanol, and then you can resuspend in a lower percentage of ethanol to store the DNA for a period of time. The inorganic procedure, again, cells are going to start in suspension. Then you're going to add some lysis buffer. If this is a red blood cell or a blood sample that we're trying to extract DNA from, we'd use a couple different types of lysis buffers. Red blood cells don't have nuclei, so those can just be burst open. White blood cells are the ones with the nuclei, so we not only have to break open the actual cell, but we also have to break open the nucleus. Then you have the lysed cells with all the cellular components, and you would do a protein precipitation. So using sodium acetate, you would then precipitate out all the protein components, which after centrifugation, those would go to the bottom, and DNA would remain in aqueous solution. Following that step, you would do the DNA precipitation using isopropanol. Solid phase, so cells in suspension. Again, we're going to want to lyse that. The supplied reagents in the kit would be used. And then you're going to have the acidification. Supplied reagents are used. DNA would remain in aqueous solution there. Then you're going to have DNA absorption under low pH. So you're going to run this through the column, then you wash it, then you run it through the column again, and you elute the DNA. In other words, the first washing doesn't wash away the DNA. The DNA remains on the solid phase. So whether it's beads or it's a membrane, it's going to remain on that solid phase. But once you elute the DNA, so you're going to add an elution buffer, elution solution, and that's actually going to flush the DNA off of the membrane or off the solid phase, and then it can be collected in a tube. And that tube can then be used for later use. Solid phase isolation media includes silica spin columns and beads. Nucleic acid binding to silica beads is the basis for many automated extraction systems. Other DNA extraction methods, limiting specimens, so fixed tissue, dried blood, bone, and rapid extraction for routine testing. The isolation of DNA from fixed tissue, preserved in fixatives is a valuable source of nucleic acid. Formalin is the most frequently used fixative. Best procedure would be acetone and 10% buffered neutral formalin. Not as good are listed below. So the tissue is embedded in parafilm, and then you have paraffin, then there's target cells within that. Then you do a microdissection. Then deparaffinize with the xylene ethanol wash, and then eventually you would digest it and put it into the buffer. Mitochondrial DNA is isolated in a similar way to nuclear DNA. We isolate the mitochondria first by centrifugation, and then you go forward with isolating the DNA. Methods to assess DNA, gel electrophoresis. So in gel electrophoresis, what we have on a typical gel, there would be a negatively charged probe on the top side and a positively charged probe on the bottom side. These are wells in which we would load our samples. So the M is for marker. This is a marker that produces bands at known distances from the top. Once we load our samples in here, the sample is going to migrate through the gel. Shorter fragments of DNA can travel a lot faster than longer segments of DNA. So this one would be the shortest segment of DNA out of the samples here. So shortest, then next shortest, then the longest. So they're going to migrate through at different speeds based on the size. So the marker here has various known base pairs or kilobase pairs in size. DNA overall has a negative charge. So when we put it in the wells here, the negatively charged electrode is here. Negative and negative repel each other. So just by electrical charge, and that's why we call it electrophoresis, the DNA is going to want to go through and migrate through the gel. And it's going to want to run toward the positive charged end, which is at this end. Sometimes we refer to this as run to red, because red is usually a positive color or a positive electrode, and black is usually the negatively charged one. So in all of our automated electrophoresis systems, that's kind of what we end up seeing. So run to red means that the DNA is going to run toward the red. So that also helps you when you're trying to put this apparatus together, that you orient the gel in the right way. Because if you make a mistake and you happen to orient it in the opposite direction, the DNA will just run off the gel, and that won't be good. So separated by size, and you can kind of evaluate DNA that way. You can also determine the concentration of DNA through spectrophotometry and through fluorometry. So RNA is going to be a little bit different. So laboratory preparation, first we have to ensure that there's a designated RNase-free area. RNases are all over the environment, and if they're all over the environment, we're going to be able to break apart the RNA. And that's not something that we want to happen. So we have to have an RNase-free environment when we're doing anything with RNA. Wear gloves, wipe up with RNase cleaning agents. Disposables certified RNase-free, rinsed in 1% diethyl procarbonate. Reagents certified RNase-free, and so forth. So again, main thing here is RNase-free environment and RNase-free reagents and disposables. The reactions, we actually want to add RNase inhibitors to it. Specimen collection, you can see there's different types of specimen collection requirements here. There are RNA-specific tubes. Some of the procedures are going to be the same. So bone marrow, we still want to, or peripheral blood, we're going to do the density gradient centrifugation or differential osmolysis. Tissue, freeze or grind, crush, and denaturant. Bacteria, fungi, plants, homogenize, vortex with glass beads. And then there's also the enzymatic digestion possibility. RNA isolation typically only has the organic or the solid phase. The organic, you're going to lyse. The lysed cells are then going to go through the extraction phase. The organic phase is going to be separated away. We have RNA in the aqueous solution, and it's going to be precipitated using ethanol. Solid phase is going to use like a membrane or beads, just like the solid phase for DNA isolation. You're going to have cells in suspension. You lyse them open. The RNA is absorbed at low pH. Wash the RNA with a wash buffer and then elute it out. Types of RNA, messenger RNA. If you recall learning about transcription and translation, messenger RNA carries that message from the DNA to a ribosome to be translated. Ribosomal RNA makes up the ribosome. Transfer RNA is going to bring amino acids into a ribosome to assist in the growing polypeptide chain. Heteronuclear RNA, when DNA is directly transcribed, an mRNA molecule is created. Now that mRNA is not ready to go right to translation. It has to be modified. So we would have to add the 5' cap, the poly A tail, remove the introns, put the exons together. So all of that processing has to happen. So heteronuclear RNA is the RNA that's made directly from the process of transcription before any post-transcriptional modifications are made. Small nuclear RNAs aid in splicing, so aid in removing those introns. Double-stranded RNA plays a role in gene silencing. So if we want to turn off certain genes, double-stranded RNA plays a role in that. And then there's many small or microRNAs have many different functions, but post-transcriptional gene regulation is one of the main ones. Isolation of mRNA. So if you remember, when we process mRNA, we're going to have that 5' cap added, the poly A tail is going to be there. So we can have a bunch of T's attached to a bead or a column, and the T's will bind to the A's from the poly A tail, and it'll capture them. So then we can put a wash buffer through it, and the mRNA will stay stuck because of the T's being present on the beater column. Electrophoresis is used as well, and then we can use spectrophotometry for getting the concentration of how much RNA we were able to isolate. Fluorometry with ribogreen can be used as well for concentration. And you can review the summary.